Evolution is one of the most powerful and unifying ideas in all of science. It explains how every living thing on Earth, from bacteria to humans, is connected by billions of years of change. When most people think of evolution, they picture fossils or ancient creatures, but evolution isn't just history. It's a measurable process driven by chemistry, genetics, and population dynamics that we can study and even observe today. In today's video, we'll explore the science behind evolution, how life began, how species change, and the forces that continue to shape every living thing on Earth. Before Darwin proposed his theory of evolution by natural selection in 1859, the story of life began much earlier, with chemistry. About 3.5 to 4 billion years ago, during the Hadean and early Archean eons, Earth looked nothing like it does today. It was hot, volcanic, and chemically dynamic. The atmosphere likely contained water vapor, methane, ammonia, carbon dioxide, nitrogen, hydrogen sulfide, and hydrogen cyanide. These gases would have been released from volcanic outgassings and interactions between magma and seawater. This early atmosphere lacked oxygen, making it reducing, which is crucial because in such conditions, organic molecules can form spontaneously when energy is supplied. And energy sources were abundant. Lightning, ultraviolet radiation from the sun, geothermal heat, and asteroid impacts all provided power for chemical reactions to occur. In 1953, Stanley Miller and Harold Urey showed that when these gases were exposed to electrical sparks simulating lightning, simple amino acids would form within hours. These would include glycine, alanine, beta-alanine, aspartic acid, and aminobutyric acid. Some scientists suggest that comets and even meteors could have delivered additional organic compounds to early Earth, while deep-sea hydrothermal vents rich in iron, nickel, and sulfide minerals may have acted as natural chemical reactors, catalyzing the creation of simple organic molecules. Once Earth's chemistry set the stage, the next big question was, how did lifeless molecules organize into living systems capable of evolving? Evidence points towards a world built around RNA, a molecule that can act both as genetic material and as a biochemical catalyst. Unlike DNA, which is stable but inert, RNA can fold into complex three-dimensional shapes that allow it to catalyze reactions, much like enzymes do. These catalytic RNAs are called ribosomes. Now, quick history lesson. In the 1980s, Thomas Cech and Sidney Altman discovered that some RNA molecules could cut and join other RNA strands without any proteins. This would earn them the 1989 Nobel Prize in Chemistry, and it proved that RNA can perform self-sustaining reactions, which is an essential step towards life. Later, researchers such as Jack Szostak showed that under the right conditions, simple organic precursors like nucleobases, sugars, and phosphates can combine to form nucleotides, which then polymerize into short RNA chains. Once some of these RNA molecules began to copy themselves imperfectly, mutations introduced variation. That variation marked the beginning of Darwinian evolution at the molecular level. Protocells, simple membrane-bound structures containing self-replicating RNA, may have been the first step towards true cells, capable of basic metabolism and growth. Now we'll talk about LUCA, or the last universal common ancestor. After countless chemical experiments by nature, one lineage of protocells gave rise to LUCA. LUCA wasn't the first living thing, but it was the ancestor from which every organism on Earth descends. By comparing modern genomes, scientists have inferred many of LUCA's traits. It would have likely used DNA for genetic storage, RNA for protein synthesis, ribosomes for translation, and ATP for energy. It relied on metal-sulfur enzymes for metabolism and probably lived in a hot, oxygen-free environment near hydrothermal vents. Once replication and variation stabilized with these early cells, biological evolution had truly begun. From this point on, populations of organisms began to change through the same processes that still operate today: mutation, recombination, natural selection, and genetic drift. Small genetic differences affected metabolism, reproduction, and survival. Over millions of years, these differences accumulated, leading to the first microbial ecosystems and eventually the astonishing diversity of life we see now. In 1859, Charles Darwin published On the Origin of Species, proposing that species were not fixed but evolved gradually through natural selection. Individuals vary within populations. Some of those variations influence survival and reproduction. Individuals with advantageous traits leave more offspring, and over generations, those traits become more common. This is what we now know as adaptation, the shaping of species by environmental pressures. When Darwin proposed his theory, he didn't know how variation occurred. Genes hadn't yet been discovered, but in 1953, James Watson, Francis Crick, Rosalind Franklin, and Maurice Wilkins revealed the structure of DNA, showing that it carries information through 4 bases: adenine, thymine, cysteine, and guanine. During DNA replication, random errors called mutations can occur: point mutations, insertions, deletions, or duplications. While many mutations are neutral or harmful, some provide beneficial traits, especially when environments change. These mutations are the raw material of evolution. Natural selection is the filter that acts on this genetic variation. A classic example comes from the Galapagos finches. When Darwin visited the islands in the 1830s, he noticed finches with different beaks, shapes, and sizes. Some thick for cracking seeds, while others were slender for catching insects. He realized they were all variations of a single ancestral species that had reached the islands from South America. Over time, differences in food sources and environments favored different traits, leading to distinct species. Natural selection had adapted them to their unique ecological niches, but evolution isn't driven by selection alone. Genetic drift refers to random fluctuations in allele frequencies, especially in small populations. For example, if a few individuals colonize an island, their gene pool might not represent the original population. This phenomenon is known as the founder effect. Gene flow is the movement of alleles between populations through migration or reproduction. It tends to reduce genetic differences between groups. And finally, mutation pressure. The steady input of new mutations adds new variation for natural selection to act upon. Now, finally, I'll be touching on speciation. Over long periods, populations can become so genetically distinct that they can no longer interbreed. This process is called speciation, and it creates a new species. Allopatric speciation occurs when populations are physically separated by a barrier, such as mountains or oceans, leading to independent evolution. Sympatric speciation happens within the same geographic area, often through genetic events like disruptive selection. At the molecular level, speciation often involves genetic incompatibilities and prevents hybrids from surviving or reproducing. Maintaining reproductive isolation is a key feature of a distinct species. From the first spark of prebiotic chemistry in Earth's ancient ocean to the rise of the first cells in the branching tree of life that eventually led to us, the story of evolution is the story of life itself, and it's still happening. We see it in viruses adapting to new hosts, bacteria evolving antibiotic resistance, and species shifting their ranges as the climate changes. The same principles that shaped Darwin's finches continue to shape life all around us and within us. I want to thank you for spending your time exploring the science of life's origins and evolution. Remember, curiosity, just like life, is always evolving.